In this video, we are briefly going to talk about the structure of the bacterial cells, their shape and size of the bacterium. In our next videos, we will talk about each of the component in detail. So starting with the shape and size of the bacteria. Bacteria are classified by shape into three basic groups: cocci, bacilli and spirochetes, as you can see in this diagram. The cocci are round, the bacilli are rods, and the spirochetes are spiral shaped. Some bacteria are variable in shape and are said to be pleomorphic. Pleo means many and morph means shapes. The shape of a bacterium is determined by its rigid cell wall. The microscopic appearance of a bacterium is one of the most important criteria used in its identification. In addition to their characteristic shapes, the arrangement of bacteria is important. For example, certain cocci occur in pairs, which are called as diplococci, some in chains, which are called as streptococci, and other in grape-like cluster called as staphylococci. These arrangements are determined by the orientation and degree of attachment of the bacteria at the time of cell division. The arrangement of rods and spirochetes is medically less important. Bacteria range in size from about 0.2 to 5 micrometer, as you can see in this graph. In this graph, sizes of representative bacteria, viruses, yeasts, protozoa and human red cells are shown. The bacteria range in size from Mycoplasma, the smallest bacteria, to the Bacillus anthracis, which is one of the largest bacteria. The viruses range from poliovirus, which is the smallest, to the pox virus, the largest. Yeasts, such as Candida albicans, are generally larger than bacteria. Protozoa have many different forms and a broad size range. Let's move toward the structure of the bacteria. The structure of a typical bacterium is illustrated in this diagram. As you can see, different structures or different components of the structure of the bacteria are labeled. First we have the capsule, then we have the cell wall, then cell membrane, then we have cytoplasm, which contains all other structures such as nucleoid, which contains the DNA. Then we have ribosomes, flagella, attachment pili, plasmids and the sex pilus. And the important features of these components are presented in this table. In this table, as you can see, the structures of the bacterium are divided into two categories, such as essential components and the non-essential components. Their chemical compositions and functions are provided in this table. First of all, we have the cell wall. The cell wall is made up of peptidoglycan, and peptidoglycan itself is made up of peptides and glycan. Glycan is a sugar backbone with peptide side chains that are cross-linked. So the function of it is to give rigid support, protect against osmotic pressure, and it is also a site of action of penicillins and cephalosporins. It is degraded by lysozyme. Lysozyme is an enzyme that is present in the human tears. Then we have outer membrane of gram-negative bacterium. Outer membrane is a component that is present only in the gram-negative bacterium, and it is present outside the peptidoglycan. It is made up of two components, lipid A and polysaccharides. Lipid A is a toxic component of the endotoxin, and the polysaccharide is a major surface antigen that is used frequently in laboratory diagnosis. The gram-positive bacteria contains surface fibers that are made up of teichoic acid, and these are the major surface antigens, but these are rarely used in laboratory diagnosis. Then we have the plasma membrane, which is also called as a cell membrane. It is made up of lipoprotein bilayer and it contains no sterols. Sterols are only present in the eukaryotic cell membranes. It is a site of oxidative enzymes and some transport enzymes. Then we have ribosomes. Ribosomes are made up of RNA and proteins, and there are two subunits of the ribosome. The larger subunit, which is a 50S, and the smaller subunit, which is a 30S. So ribosomes are the site of protein synthesis. Moreover, they are the site of action of some antibiotics such as aminoglycosides, erythromycins, tetracyclines, and chloramphenicol. Then we have nucleoid. Nucleoid is the region in the bacterial cell cytoplasm which contains the DNA of the bacterium. Then we have mesosomes. Mesosomes are the invaginations of the plasma membrane of the bacterium into the cytoplasm, and these contain some secretory enzymes, and it also helps in the cell division of the bacterium. Periplasm. Periplasm is a space that is present between the plasma membrane and the outer membrane, and it is only present in the gram-negative bacteria. It contains many hydrolytic enzymes including beta-lactamases that degrade the beta-lactam drugs. Now we have the non-essential components. First of all, we have capsule, which is made up of polysaccharides and it protects against the phagocytosis. Then we have pilus or fimbriae, which is made up of glycoproteins. There are two types of pili. First type is that mediates in attachment of the bacterial cell to surfaces. Then we have the second type, which is the sex pilus, which mediates attachment of the two bacterium during conjugation. Then we have flagellum, which is made up of a protein called flagellin and it provides motility to the bacterial cells. Then we have spores. Spores are keratin-like coats which are made up of dipicolinic acid. These spores provide resistance to dehydration, heat and chemicals. Plasmids. Plasmids are extra chromosomal DNA molecules which contain a variety of genes for antibiotic resistance, heavy metal resistance, and they also contain genes for toxin production. Then we have granules. Granules are made up of glycogen, lipids and polyphosphates. These are site of nutrients in cytoplasm. At last we have glycocalyx, which is made up of polysaccharide and it mediates adherence to the surfaces.